Hi everybody, my name is Natalie Villafranca, and today I'm going to be talking about severe acute respiratory syndrome, also known as SARS, also known as SARS-CoV. So its taxonomy, the domain is virus, its phylum is Pisuviricoda, its class is Pisoniviridae, its order is Nidovirales, its family is Coronaviridae, its genus is Beta coronavirus, and its strain is SARS-CoV, also known as severe acute respiratory syndrome related coronavirus. So, characteristics of the infectious agent, it has a very large genome compared to other positive sense RNA viruses. 10 genes in it modulate innate immunity. It's positive sense, single-stranded with a 5' cap. It is 26 to 32 kilobytes of genomic data. Its structure is a spherical enveloped virus. The lipid bilayer contains glycoprotein spikes and holds the envelope, nucleocapsid, and membrane proteins. There are 3 notable coronavirus strains: SARS, MERS, and COVID-19. The SARS epidemic occurred in 2002, the MERS epidemic in 2012, and COVID-19 pandemic this year. So pathogenesis and immune response. The pathogenesis of SARS is partially controlled by, is controlled by the innate immune system. The S protein has an ACE2 receptor, which will attach the virus to the cell membrane, and then endocytosis will occur via transformation of the S1 and S2 region of the S protein, which will then release its domain and expose its fusion peptides. The rearrangement of the S protein will allow for fusion after the S1 domain has been shed. It is hypothesized that viral replication occurs via autophagy, which is where cytoplasmic proteins create isolation membranes that surround material that'll be degraded. In terms of its immune response, in viral infections, the expression of antiviral cytokine interferon is an indicator of the adaptive immune response working to get rid of the virus. But something interesting about SARS is that it did not include this interferon, which means that SARS is actually an innate immune system virus. It is maintained by different cells in the airway epithelium, which include dendritic cells, innate lymphoid cells, and alveolar macrophages. The beginning of the body's response occurs via pattern recognition receptors, which will then recognize the pathogen-associated molecular patterns of the virus, so like the PAMPs. And after the PAMPs go through recognition, 2 types of interferons and other cytokines will go through transcription. Autocrine and paracrine signals found in infected and uninfected cells will assist in the translation of cytokines, which will assist in the immune response. So in this chart right here, you can see like the viral replication cycle that we learned in class. So the virus will bind to the receptor, and then viral entry, membrane fusion, and then that viral RNA, there's actually a typo in that, the viral RNA will release its genomic RNA, blocking, and then the RNA will go through replication, and then transcription, and then you'll get your viral RNA in your cell, and then there will be assembly and budding, and then that mature viron formation, and then you'll have that virus release into your cell. So pathology, this is a photo taken from a paper that shows 4 different tissue sections of lungs from mice. The first section, I don't know if this is showing my mouse, but the first section on the top left is an uninfected mice. The second is a mice infected with the wild type SARS-CoV, and the bottom 2 with either functional PDZ binding motif or, PBM binding motif, or no PDM binding motif. So we can see that the mice with no PBM protein had clear functional airways, and as did the non-infected. And if you look at the two on the right, the SARS-CoV wild type and the SARS-CoV PBM positive, the those two had collapsed airways, which led to lung failure and death. So symptoms, so pretty similar to COVID-19, flu-like symptoms like cough, sore throat, fever. Most experienced shortness of breath as well, which would progress, but then this can progress into pneumonia. So SARS can also progress into ARDS, which is acute respiratory distress syndrome, which during the epidemic, about 1/4 of the cases went into ARDS from SARS. SARS to ARDS, and had a mortality rate of over 50% once you went into ARDS. So about 20 to 30% of patients required ventilators during the epidemic. And so ARDS is associated with the activation of cytokines involved in inflammation response. There was a really high amount of expression of these cytokines in the lungs of SARS patients. In viral infections, the expression of the antiviral cytokine interferon is an indicator of the adaptive immune response working to get rid of the virus. So diagnosis. There's not really a good diagnostic test available during the epidemic. They tried using qPCR, but it didn't really work very well. So the peak detection rate was really, really low. So this is like a picture from a qPCR, and like these peaks are where it shows like how much of that like gene is being expressed, or like how much of the virus is being expressed. So the peak detection rate was using respiratory specimens, it was 2 weeks, and could even be at the 4th week if they were using urine. So the best method that they had was using tracheal aspirate and stool samples, and it had a higher diagnostic rate, but it was still only about 60%. So there were a lot of negatives, even though they were positive. And a lot of the times when they would test, like the only time you would get a positive is like if you were dead or like very, very ill, which means that like you would need a lot of the virus to make you really, really sick. So transmission. Direct contact, respiratory droplets, and touching your face after not washing your hands. So direct contact would be having bodily fluids of an infected person come inside a healthy person, or respiratory droplets, which is the reason we're wearing masks now. When people like sneeze and cough and like even talk, like those droplets will project out, and they can project out up to like 3 feet. So that's a way of transmission. And touching your eyes and mouth and nose after like touching door handles that other people have touched is not good. So kind of the history of the epidemic. So the SARS pandemic of 2002 began in the Guangdong province of South China. The disease spread to other Southeast Asian countries, with most of the cases centered in Singapore, Hong Kong, Taiwan, and Canada. Here I have a timeline that shows the timeline of the epidemic. So November of 2002, the first case of an atypical pneumonia was reported in Guangdong. And March 15th, which is about 4 months later, the CDC issued their first health alert, hosting a media telebriefing. So March 24th, about 10 days later, the CDC lab suggested that the disease agent could be a new coronavirus, and there were about 39 suspect cases in the U.S. I'm moving myself again. And May 6th, so about 2 months later, there were no new probable cases of SARS in the U.S., which means that the U.S.'s containment was successful. So the WHO announced that the SARS outbreak was contained on July 5th, 2003. So less than a year after it had begun in Guangdong. So for the prevalence, this photo shows a map of all 8,096 cases, of which 774 resulted in death. So that's about a 10% mortality rate in comparison with SARS-CoV-2's 2.5 mortality rate. The fatality rate among people under 60 was 6.8%, while the mortality rate in older patients was about 43%. So most of the cases were in China, Hong Kong, Taiwan, and Singapore. And then a few had spread to Europe, and even more spread to Canada and the US. So treatment, on a paper that I read, it said the best treatment strategy for SARS is still unknown, but these are some of the ones that they were using. So ribavirin is used to treat respiratory syncytial virus infection. Corticosteroids can lower fever and show changes in a chest radiograph, but they stimulate viral replication. And then convalescent plasma, which is pretty similar to what we're doing now. So like when people get sick and they have their plasma and then they donate it to sick people, that could be beneficial. Non-invasive positive pressure ventilation, so things like a CPAP. And then traditional Chinese medicine, I thought that was really interesting that a scientific paper would suggest that, because usually like Eastern and Western medicine don't usually mix. So at-risk populations, the virus can infect everybody, but people who were elderly and older than 65 had a 50% mortality rate. So anyone who had been in close contact with an infected person could also be infected. Prevention, there hasn't been any cases of SARS since 2004. It's contained. It's like not existing anymore. But similar to COVID-19, ways to prevent getting it are washing your hands, wearing a mask, don't mess with nature so you don't get zoonotic disease outbreaks. I wanted to do this comparison really quickly because I thought it was really interesting and could be like a point of discussion for class. But SARS-CoV versus SARS-CoV-2. So like a lot of the times we hear in like the media talking about like SARS-CoV-2, the virus that causes COVID-19, right? Like the disease. SARS-CoV had over 8,000 cases, killed over 800 people, and then R0 was 0.5. It was in 32 countries and originated in Guangdong. While coronavirus, the pandemic that we're living through now, there are over 55.6 million cases and have killed over 1.34 million people. So in the US alone, there's been 12.2 million cases and 250,000, well, more than 250,000 dead. And so our R0 for COVID-19 is actually 2 to 3. And it's in 218 countries and territories and originated in Wuhan. So this is kind of interesting because they're from the same family. So why is COVID-19 so much more prevalent? It's pretty interesting because SARS was actually a lot more deadly with about 10% mortality rate, while COVID-19 has a like 2.5% death rate. So that's pretty interesting. Another really interesting thing to think about is how COVID-19 originated in Wuhan, which is one of the most populated places in China. And so people were traveling in and out, and it took like forever to realize that it was even spreading, which is why it spread so much more. And then also, once it had already spread, hospitals were out of beds. So people were like told to like go home, and like therefore like we're out in public more, and like we're having more contact with people. And so that's pretty interesting to consider. And then another thing is that COVID-19 actually has asymptomatic carriers that are infectious. So like if you're asymptomatic, you can get someone else sick. So a lot of people didn't actually know that they were sick, whereas like SARS, there were no asymptomatic cases. So this could be an interesting like point of discussion. So research efforts, when I was looking up the literature, it was all taken over by COVID-19. But going off of this presentation, I can say that there should be better testing that can actually detect the virus. I don't really know what other things we could use besides qPCR, because we're using qPCR now for this virus. And then also research on treatment to better understand how to treat SARS. So thanks for watching, and I look forward to seeing you on Tuesday.